seminar. I want to share with you some of the results of my dissertation research. And the title of my dissertation is Ecological Drivers of Range Limitation in Species. But before I dive into my talk, I want to spend a moment and say thank you to Oru Gawe, who is my PhD advisor, who has been very supportive during my time here and who was a good mentor for me. I want to thank my committee members, Stephanie Kivlen, Laura Rousseau, Charles Quitt, and Jim Fordyce for providing guidance and helping me to shape my dissertation. Rob was a very good friend of mine. He helped with my coding. Lalasha, Hyler, Ben, Taylor helped and give me feedbacks on my manuscript. Diane, Angela, Lucas, Atma, Bruno, Lona helped with my integration here in the US. I want to thank the Bailey, Schweitzer, and Kivlin Lab for allowing me to attend to the lab meeting and receive comments from the students. ITA for Jim Fordyce, Jessica Birdke, Ed Schilling, Paul Amshoff, and I learned a lot from them. I want to thank my lab mates, Lauren and Nadeja, my field assistant, Deo, Herman, Christian, Saintish, and all these institutions for giving me money to do my research. Every species on Earth has limit to its geographic distribution, and some species have a wide range, while other species have a restricted range. Over the past 4 years, I've been interested in understanding what mechanisms drive range limitation. And that question is very important because being able to predict what is happening here at the periphery, for example, if a species is getting extinct or adapt, can help us to anticipate the effect of climate change and human disturbance on range-limited species. One of the most used hypotheses to explain range limitation is the abundance center hypothesis. And that hypothesis was proposed in 1984 by James Brown in a paper published in American Naturalist. And he published this map. The map is the variation of the density of this bird. And the intersection line between the vertical and the horizontal line represent the center of the range, and you have the periphery here. He made an observation that the average density decreased from center to periphery. And based on this species and based on a bunch of other species, he concluded that in general, species have a higher abundance at the center of the range and decline in abundance toward the edge. So in addition to decrease in abundance from center to periphery, many authors began reporting decrease in population growth rate and genetic diversity from center to periphery. And this is called the center-periphery hypothesis. However, a recent study published by Dallas and colleagues showed that species are not most abundant at the center of the range. So whether it is the geographic range or the climatic niche, species are not most abundant. And this was the first paper to be conducted across taxa and using more than 100 species. And as we can see, there is no relationship between abundance and geographic or climatic distance here. So as soon as the paper got published, the paper received a bunch of critics, and two comments were written at the same time. One by Jason Roof, who is at St. Louis, Missouri, and the second one from Mona, PhD advisor Peterson. And the takeaway from these two comments is that the data that were used by Dallas and colleagues were not appropriate to test the center-periphery hypothesis, and B, the estimation of the distance from niche center was not accurate. So there is this debate going on the center-periphery hypothesis, and the first part of my dissertation want to state a position in that debate. And I'm asking here, are species most abundant demographically most performant and genetically most diverse at the center of the range. And to do that, I did a global synthesis on studies conducted over the past 50 years. So to gain a mechanistic understanding of the drivers of range limitation, I also use a case study system, a study system in Africa to investigate how biotic and abiotic factor could affect species abundance from center to periphery, and what is the role of herbivory from center to periphery. And finally, how demographic parameters vary from center to periphery. So I'm going to talk about all these questions during my talk. Let's go back to the first question. Are species most abundant, demographically most performant, and genetically most diverse at the center of the range? I predict that, yes, species are most abundant, demographically more performant, and genetically more diverse at the center of the range than at periphery. And to test this hypothesis, I conducted a meta-analysis using study testing the center-periphery over the past 50 years. For each study, I estimate the effect size. Here, I prefer the HST. And the X here represent either population abundance, population growth rate, or genetic diversity. So C represents those values for central population, and P represents the peripheral value. And the S here represents the pool standard deviation. The J represents the sample bias correction factor. Because each study has a different sample size, I estimate also the precision, which is just 1 over the variance for each study. And I use those two metrics to estimate the cumulative effect size across the whole studies. And we have support for the center-periphery hypothesis only if D here is significantly higher than 0. We did that and we found partial support for the center-periphery hypothesis. On this graph, you have on the left side all the response variables that we include here in our meta-analysis: population growth rate, genetic diversity, and abundance. For each response variable, we provide the cumulative effect size here with a confidence interval. And we provide also the number of study, the K here, the number of study that we use to test if we have support or not. The dashed line here represents where there is no difference between central and peripheral population. As you can see, we have support only when we use genetic diversity as response variable, suggesting that in general, species have higher genetic diversity at the center than at periphery. The extent to which the effect size vary across a meta-analysis is called a between study heterogeneity. And this is the I-square here. And those values are more than 60, suggesting that we have high heterogeneity, and we have a large variance in our results. So another question to ask here is, why do we have such a large variance for study using the same response variable? So as we were trying to understand that question, we realized that people use two types of distance when they test the center-periphery hypothesis. The first one is distance from geographic center. And that distance is estimated across the geographic range of the species. So you have here the coordinate of each population. That blue star represents the center, and you have the periphery here in red. So the distance from geographic center is just the distance between this yellow dot and each of these yellow dot and that star, that blue star. And the second distance is the distance from climatic center. And when we talk about distance from climatic center, we have the Euclidean distance and the Mahalanobis distance. And this instead of having the coordinate of each population, this is done across the climatic variable. As you can see here, we have precipitation and temperature. And this blue star represents the center of the climatic niche. And the Euclidean distance here will be just the distance between each yellow dot and the blue star. Now, the difference between the Euclidean distance and the Mahalanobis distance is that the Mahalanobis distance take into account the correlation between the variable. For example, precipitation and temperature take into account the correlation. That's just the difference. So here we predict that study using distance from niche center, we found support for the center-periphery hypothesis, and studies using distance from geographic center, we not found support for the center-periphery hypothesis. We did that, and we found that using the two climatic distance, which is the Mahalanobis distance, the Euclidean distance, the study using those distance did not find support for the center-periphery hypothesis, but studies using geographic distance found support for the center-periphery hypothesis. And there are two things that can explain this result. First, the way we estimate the climatic niche is not well done, is not appropriate. And second, there is probably a correlation between, a spatial correlation between the geographic range and the climatic niche for the study we use to test the effect of geographic distance on effect size here. The takeaway from this part is that species are not most abundant or demographically most performant, but they are genetically most diverse at the center of the range. Depending on the method study used, they can find also support or not for the center-periphery hypothesis. We found that when the study used geographic distance, the study is more likely to find support for the center-periphery hypothesis. So what is the role of biotic and abiotic factor in shaping species range limitation? So to test this, or to answer this question, I use a case study, a tropical system. And one of the reasons I chose a tropical system was this map. And this map is just a result of the meta-analysis I just showed you. As you can see here, the number of studies testing the center-periphery hypothesis in tropical region is very low compared to the number of studies testing out of the tropical region. So there is a bias toward temperate region. And that was one of the motivation of one of the motivation or the reason why I did that study. So I went back to Africa. And I went to this country, Benin, where I was born and raised. And I sampled 12 population of this species to measure attaquensis, which is the perennial herb, attacked by two herbivores. The first one is the larvae of this butterfly, and the second one is this beetle. And this is a very good system because the perennial herb allow us to understand the demographic response over a short term. It's not like a big tree, we have to wait 10 years before having a response. And second, we have this herbivory rate that we can use to understand ecological interaction from center to periphery. So that was a good system to test the center-periphery hypothesis. We also estimate the distance from niche center. And to do that, we estimate the climatic niche using climatic variable. And we estimate, and this is the representation here, and we use the climatic niche to estimate the distance from climatic center. We also estimate the distance from geographic center using the geographic range of the species. And we test if there is a correlation between these two distances, and we found no correlation. between the two distances. Here I predict that abiotic and biotic factor mediate the relationship between distance from range center and population abundance. To test this hypothesis, I built the structural equation model. And here, distance from niche center could affect species density directly here via a reduction of climatic variable from center to periphery. And distance from niche center could also affect species density via a decrease of abiotic factors. And the beta 1, beta 2, beta 3 represent just the strength of the relationship here. We have a mediation effect, or the mediation effect is stronger than the direct effect if the product of beta 1 and beta 2 is more than beta 3. So we use this, and we found that there is no relationship between climatic distance and species density. As you can see, the dash line here represents the relationship for which we fail to find support, significant relationship, sorry. So climatic distance did not affect species density, and abiotic factors such as soil nitrogen, soil pH, soil moisture, and light did not decrease also from center to periphery. As you can see, there is no effect of climatic distance on this variable. Species competition here, the competition between plants in each of our plot did not affect species density as well. Species density was mainly driven by soil nitrogen and soil potassium. And soil potassium was also driven by altitude. So we conclude that abiotic factor mediates the effect of altitude on population abundance. For this part, we use species competition as the only biotic factor. So what about herbivory rate? What is the role of herbivory in driving species range? Previous studies show that high herbivory rate at the periphery drive a decrease in species abundance from center to periphery. But this is not the case for species that are herbivory tolerant, for example. We test that to measure in our study system to measure occurrences. And we predict that abiotic and biotic factor mediate the effect of climatic or geographic distance on herbivory rate. To test that, we use again a structural equation model where we predict that those abiotic factors such as soil phosphorus, soil moisture, soil nitrogen, soil pH, soil potassium, and light will decrease from center to periphery because we assume that the center of the niche has a better condition for the species. And then we predict that herbivory rate will increase with an increase of abiotic factor consistent with the resource availability hypothesis. And as phylogenetic diversity increase, we assume that herbivory rate will decrease. We did that and we found that herbivory rate decreased from center to periphery. And this contrasts with what we predict early. Soil nitrogen decrease as well from center to periphery, and herbivory rates increase with soil nitrogen, suggesting that there is a mediation effect again of soil nitrogen on herbivory rates into measure population. Now, my last question is to understand or to ask how do demographic parameters vary from center to periphery. And so far, I use species abundance. And when we use species abundance, we just have a snapshot of what is happening at the population level. It doesn't tell us if the population will increase or decrease over the long term. So in this part, I propose to use population growth rate, which is a more reliable metric to understand if the population will increase or will decrease, and tells us about the health of the population in general. Population growth rate also takes into account life history trade-off, so it's a very good metric to understand range limitation. Here I predict that as ecological condition worsen at the periphery, demographic parameter will decrease and thereby drive range limitation. And the first demographic parameter I used here was vital rate. And by vital rate, I mean survival rate, reproduction, and growth. And because I assume that the center of the climatic niche will have better condition again, I predict that the vital rate will decrease from center to periphery. And then across the geographic range, I do not expect any significant change of vital rate from center to periphery. To test that, I use demographic data collected on my study system again, Tumbeja. So in 2019, I went back in Africa and I tagged all the individual of Tumbeja Taquensis. You can see here the number. And what I did over 3 years, I went back at the same time to collect the same data. For example, survival, to see if the species is still there or not. And the number of flower, I just count and note, and then the number of fruit. So I use that as demographic data to see if the population will increase or decrease over the long term. What we found here is that the probability of flowering and giving fruit does not change significantly across the climatic niche, as you can see here on the left side of the graph. However, the probability of flowering increase at the periphery of the geographic range. For the probability of producing fruit, we found also no significant relationship between that probability and the geographic distance. Similarly, for across the climatic niche, the probability of growth and the probability of survival did not change significantly from climatic center to periphery, as you can see here, but the probability of growth increased significantly from geographic range to geographic periphery. And survival rates also did not change significantly from geographic center to periphery. So the second and the third demographic parameter I use here was population growth rate. I predict that population growth rate will decrease as well from center to periphery across the climatic niche, and population growth rate will not change significantly across the geographic range. I predict the same thing for the damping ratio. Now, the damping ratio is a proxy of how a population recover, how fast a population recover after perturbation. So it's a metric of the resilience of a population. To test those two hypotheses, I use this formula, which is an integral projection model, where the size of the population at t plus 1 is a function of the size of the population at t. And the probability of each individual in the population to survive and grow, or survive and give new offspring here. Here, when we use this formula to estimate the population growth rate that we usually call lambda. Here, when lambda is more than 1, the population is projected to increase over the long term. When lambda is equal to 1, the population is projected to be constant over the long term. And when lambda is less than 1, the population is projected to decrease over the long term. Again, we found no relationship between population growth rate and geographic distance or climatic distance. However, we found that as nitrogen in soil increase, population growth rate decrease. We found also no relationship between population growth rate and soil phosphorus. This relationship between population growth rate and soil nitrogen could be explained by herbivory rate again, because we previously showed that soil nitrogen drive herbivory rate into measure at appearances population. So here maybe herbivory rate is a mediation factor, is playing a mediation role between population growth rate and soil nitrogen. And to test that, we need to use herbivory rate as a covariate in our integral projection model to see how an increase of herbivory rate will decrease or increase population growth rate. Finally, we found that across the climatic niche, we have no significant relationship between how fast the population recover after perturbation and distance from climatic center. And we found that population resilience to perturbation decrease across the geographic range. Here, the takeaway from this part is that population are demographically more resilient at the center of the geographic range than at the periphery. To wrap up now, we asked 4 questions. The first question was, are species most abundant demographically, most performant and genetically most diverse at the center of their range? And to answer that question, we use a global synthesis. And we found that species are not most abundant, demographically most performant, but they are genetically more diverse at the center of their range. We also found that depending on the method the study used to test the center-periphery, one could find support or not for the center-periphery hypothesis. Then our second question was how biotic and abiotic factor affect species abundance from center to periphery. And we found that climatic variable does not really play a big role in driving species range, and rather species abundance was driven by local factors such as soil property. Our third question was the role of herbivory from center to periphery, and we found that herbivory rate decreased from center to periphery, and that mediation role is done by soil nitrogen. The last question was how do demographic parameters vary from center to periphery, and we found that central population are demographically more resilient to perturbation than peripheral population. Thank you. I will take questions.
No, you're good. Okay. I'm going to get close to Jacob here. So for those on Zoom, if you want to ask a question, feel free to join and do that by the chat. I'll keep a monitor of that. And Oru is going to keep an eye on the room here and monitor the Q&A here. So, Evan.
Any question for Jacob?
Yeah.
First of all, that was great. Congratulations.
Thank you.
The question is, do you have sufficient data to say whether there's any phylogenetic component to this, the differences between clusters of species?
Yeah, thank you. Yeah, we have enough data. We have probably more than 200 species, and we test that, but I did not show that here, to see if close related species behave the same way from center to periphery. And we found no significant phylogenetic signal when we did that. So there you see species that are very close, but they behave differently from center to periphery. Yeah.
All right. I've got two questions.
Okay.
Do people ever try to apply these ideas to marine?
Yes. Dallas did that. I did that in the paper in Ecology, and that was one of the critique by Jason Knopf, and he said that fish are not appropriate to test the center-periphery hypothesis. So you see some studies, using fish system to do that, but it's not common. People prefer mammal, bird, or plant. Yes.
So was your confidence or your uncertainty in all those points of similar size, or was there variation in that, and then did you do a weighted regression?
Oh, I did just a standard regression. But for the meta-analysis, I weight for each studies, and the weight was the precision 1 over the variance of each study. It was not constant across all the studies.
Sometimes you have things where it looks like there was a trend, but then you have one outlier.
Yeah.
The question in my mind is, how confident are you?
Yeah, I think there are two. You can use the Cook distance, for example. A Cook distance, and that's a statistical method to know if the outlier affect or not the regression. And in our case, those outliers did not affect really the regression. Yeah. Yes. Yeah, I use animal and plant, and I found a difference, but I did not also present that. They found support for plant, and we did not find support for animals. Yeah.
Question?
Yeah, Joe.
Before I ask my question, I just want to say thank you for choosing this department. A personal pleasure to have you in that group and be part of this community. I have to see you. I have two questions. My first one is, what is your hypothesis for why resiliency is greater in the center of the geographic range? What's driving that?
I think the main idea behind population at the center being more resilient or being more increasing over time than peripheral population is just we assume that the center of the climatic niche has often the better condition for the species in terms of abiotic and biotic condition. And yes, you said? Yes, I did not find that across the climatic niche, but we found that for across the geographic range. So maybe population at the center of the geographic range has better climatic condition than population at the periphery range. That's one reason. But yeah, we need to dig deeper and see why we have that. Using the damping ratio and use more species could be something very interesting to look at in the future, particularly as climate change is coming, is recurrent, is going to be something very interesting to understand.
Before I actually get, if you don't mind, I'll ask just a quick follow-up question on Joe's question, which I found really interesting. So because there's a mismatch between climatic distance and geographic distance, would you say that some other factors any
Yeah.
other factor could?
Yeah, there are many factors that could come to play here, but I try my best not generalize from just my one species. So that's why I was saying that, you know, if we can have more species, they have this database, the compadre of IPM, Padre, I don't remember exactly the name, Padrino, which is just a big data set of species, and you can use IPM also. So if we do that for all the species and we find the same pattern, that's going to be something interesting. But just trying to generalize from our own study from somewhere, you know, in Africa is a bit risky.
So I have a Zoom question.
Yes.
I'm up close to you here, Jacob. This is coming from Hyler. Hyler's in the audience on Zoom, one of our former undergraduate great students here. If this is really truly who this is coming from. Jacob, in your opinion, what is the future of center-periphery hypothesis research? That's a loaded question there. What are the most important avenues that need to be next explored to better understand range-limited plant species and how they behave?
Wow, that's a good question. I think we need to continue to test that hypothesis. It's just the way we do that now. The way we do that now can be improved. For example, when we estimate the climatic niche, most of our model do not incorporate biotic factors. We just use climatic variable, precipitation, temperature, and all this stuff. And for some species, biotic factors are very important drivers. So I think if we can improve our model, this will help a lot to use the center-periphery hypothesis to understand the range limitation.
Yeah. One last question before.
So you found in your meta-analysis that the higher genetic diversity in the center.
Yeah.
Was that true of your model species? And did that explain your increased resilience to disturbance?
That's possible, but I did not test any genetic variable using my system. I just did abundance, herbivory rate, and population growth rate. My background is not genetic, but I'm working on it to understand how genetic diversity vary using my study system. Thank you.
Well, thank you very much for those great questions from Jacob.
Just so people know, two parting thoughts for those who want to continue discussing the things that were discussed here in the seminar. Feel free to go to Public House and continue that conversation. But most of all, have a happy Thanksgiving, everyone. Be safe and stay healthy, and we'll see you in 2 weeks.